It's my pleasure to introduce a friend and longstanding colleague, Dr. Chris Hunter, who's going to give the first keynote talk of today. So Professor Hunter obtained his PhD at the University of Glasgow in the UK and undertook a postdoctoral fellowship at Glasgow, where he developed his interest in the role of cytokines in immunity in the central nervous system. This was followed by another postdoc at Stanford before he joined the faculty at the University of Pennsylvania. And there, his research group continues to study cytokines that influence both innate and adaptive immune responses to infection. His collaborative studies with the academic and biotech communities over the last couple of decades on IL-12 family members has helped to define their role in the induction of cell-mediated immunity that have been actually quite broadly relevant to many types of inflammatory processes. For example, his discovery that IL-27 was a potent inhibitor of cell-mediated immunity has formed the basis for work in its therapeutic potential in cancer and in autoimmunity. He has been a journal editor and member of various editorial boards, as well as being involved in extensive reviews. He's a past president of the International Cytokine and Interferon Society and served on other departmental committees. He's currently the Mindy Halleckman Higher President's Distinguished Chair and Director of the Institute for Infection and Zoonotic Diseases. And without further delay, I'd like to hand over to Chris. And Chris, would be delighted if you'd please go ahead with your presentation.
Thank you very much, Eleanor. It's been a pleasure this morning listening to some of the talks. Clearly a very high level of science, and it's a real pleasure to be asked to participate today. I have a very simple story to tell, or as an immunologist, I think it's simple, and I hope that everybody can follow along. I'd like to start with sort of acknowledging the individuals who did the work. So the talk today will be about how mRNA vaccines promote CD8 T cell responses. And this is a project that's been ongoing really for over 4 or 5 years. And the work has been done, I hope you can see my cursor, by Mohammed, Emily, who's a graduate student in the lab, and Anthony, who's a research fellow in the lab. This work has all been done in collaboration with Drew Weissman here at the University of Pennsylvania, and that's shown here, and is supported by the Bassett Center for Cancer Research, the nanomedicine core at the School of Medicine, research funds from Penn Vet, but also just to highlight, perhaps in a topical fashion, that the work is supported by the National Institute of Health. And I think what you're going to see is, basic science work that supported by the NIH that can translate into, new therapies for different forms of cancer and infectious diseases. Just to kind of run ourselves through the sort of principles of things I'm going to talk about, I'm just going to outline the basic cell types that we're interested in and some of the biology we're interested in, and then going to try and sort of select certain data sets that I think illustrate the principles that we're interested in understanding. I don't have time to show every piece of data in depth, but I'm just going to try and focus on some key data sets that shaped how we think about mRNA vaccines. I'm going to start with this sort of like little illustration of the T cell that we're most interested in the lab at the moment for this particular project, and that's a CD8 T cell. And this cartoon is from a delightful manga series called Cells at Work, which describes cells of the immune system. And for those of you that haven't seen it, I really suggest that you go and take a little look at it. And it talks about the killer T cell, the CD8 killer T cell. It's a white blood cell that recognizes and kills various foreign matter and unhealthy cells. I love the description that he is aggressive, cocky, and rowdy, brutally taking pleasure in slaughtering pathogens and unhealthy cells. The cell that I'm going to really sort of focus on generating and thinking about is the CD8 T cell. And the other slide, which is taken from Wikipedia's section on cytotoxic T cell, just shows a tumor cell in the middle surrounded by 3 cytotoxic T cells. And these CD8 T cells are important for a range of resistance to a range of viral, bacterial, and parasitic pathogens. They're also important for tumor surveillance, and I'm going to try and perhaps illustrate some of that as we go through this. And really, our goal is to be able to take this cell that's part of our natural adaptive immune system and think about ways of harnessing them in the context of vaccination. And then the next slide will just be a little reminder of how lipid nanoparticle deliveries of mRNA, pseudouridine modified mRNA works. So essentially, I think many people on this, unless you've been in a coma for the last 4 years or 5 years, we should all be aware of the use of lipid nanoparticle delivery systems for mRNA, for COVID, and the spike specific vaccine that's used to protect individuals from COVID infection. And these lipid nanoparticles are illustrated here. And they're not, they're really a sort of very dense particle that contains mRNA inside this particle. It encapsulates the mRNA. And the mRNA can code for a variety of different proteins. The one we're most familiar with is the spike protein it's encoded for by COVID. This mRNA is modified. The pseudouridine modification makes this mRNA less pathogenic, less inflammatory. And that's one of the keys to the success of this particular vaccine. But when these lipid nanoparticles are injected into us or given to a cell, there are various receptors on the surface of these cells that lead to their endocytosis, leads to their fusion with the endosomal membrane, the low pH, and the release of the mRNA into the cytosol of the cell that's taken up this particle. This mRNA, of course, is translated and a protein is made. And then depending on how you modify that protein or how you've encoded that protein, it can be directed to the surface of the cell, or it can be secreted, or it can be maintained within the cell. And for the spike protein, for example, it's put on the surface of the cells that have taken up this mRNA, and then that leads to the induction of a really potent immune response. Most of us will be aware that, the focus of this vaccine initially was the ability to generate antibody responses, and that's a classic criteria for the ways of assessing vaccine success. But the other thing that this mRNA vaccine does is that it's really, really good at generating a CD8 T cell response. And this next slide just illustrates what happens in an individual that is vaccinated with the COVID mRNA vaccine. I suspect anybody that's listening today or still listening today has either encountered COVID or has been vaccinated with the COVID mRNA vaccine or some sort of vaccine. And essentially what we're looking at here is a flow cytometry plot using 2 tetramers specific for the spike protein. So essentially this is just a way of trying to identify CD8 T cells that would be induced in response to vaccination or infection. So what we have here is an individual who was bled at day 0 before being vaccinated and then given various doses of vaccine. I think I've now had 4 doses of the vaccine. I'm actually part of this study as well, although these are not my T cells. And in an unvaccinated individual who's not been infected, there are no spike-specific CD8 T cells. After a primary vaccination, here you find these spike-specific CD8 T cells. And then after a second dose, you sort of enhance this response, right? So you go from 0 to a low frequency to a much higher frequency. And then as that dose, as time goes past and you generate memory cells, you're left with these CD8 T cell memory cells. This is baseline. And then after the third dose, you again see this sort of peak of the CD8 T cell response. And for any of the immunologists who are listening, this is how we think about the immune response. You encounter a pathogen, you generate a T cell response, you encounter it again, you generate more of those T cells. So we're sort of really interested in thinking about, well, how does this work? A large number of patients, of individuals on this planet have been vaccinated with this vaccine. We know it generates really good antibody responses, but we don't really understand how you get these great CD8 T cell responses. And so one of our basic questions is just to try and think a little bit about how does that work? And so just to sort of like wind ourselves back in time a little bit, the way we would think normally about how CD8 T cell responses are generated is kind of illustrated here. In response to a pathogen, like a bacteria or a parasite or a virus, cells like dendritic cells encounter these pathogens and they present antigen. And if the CD8 T cell sees its right peptide in the right context of the right MHC, these CD8 T cells will expand and help to control the infection. And this is really sort of what we refer to as signal one, and that leads to T cell activation. But there are other signals mediated by cytokines, in particular IL-12. It's a heterodimeric cytokine composed of these two subunits here. And this is really one of the major cytokines that we think of as being able to drive CD8 T cell expansion. The other side, and of course, 12 is not the only cytokine that does this, but there's a literature that other cytokines like IL-6 and type 1 interferons can also promote CD8 T cell responses. So it seemed like a reasonable place to start to think a little bit about, what cytokines are produced during vaccination that might influence these CD8 T cell responses. So in the next slide, you can see a sort of a very simple assay where essentially, mice have been vaccinated with lipid nanoparticles. These lipid nanoparticles are put in intramuscularly. They then flow into the lymph node. And when they're there, they interact with macrophages and monocytes and dendritic cell populations. And this leads to the production of cytokines. So these lipid nanoparticles and the mRNA are not silent. As for all of us who've had this vaccination, we know they're not silent. We feel the immune system kicking in once we get vaccinated. And all we've done here is take the lymph nodes, the draining lymph nodes, grind them up, and at 6 hours post-vaccination, simply looked for the levels of cytokines that were induced locally within this lymph node using a Luminex assay. And this sort of heat map just illustrates those cytokines that are increased in response to vaccination. And the top cytokine, the starred cytokines are factors that are usually produced by monocytes and macrophages and dendritic cells. And IL-6 is what is the top cytokine that's induced. And we know that this is really important for humoral immunity. There's another cytokine called IL-27 that's here, but here's IL-12. That's induced approximately twofold. And then the type 1 interferons are down here, and they're sort of induced to similar levels. So the cytokines I'm going to focus on are shown here are IL-12 and 27 and IL-6. And these cytokines, again, there are many reasons people don't like immunologists. And one of them is that this nomenclature does not tell us anything about their relationship between 6, 27, and 12. And in fact, these are cytokines that are very closely related. They're structurally related proteins. They use similar subunits. So for example, receptor subunits, for example, IL-6 uses this cytokine subunit GP130, and that's shared by IL-27. IL-12 and 23 are very closely related. They use similar cytokine subunits and have distinct subunits as well. They share receptor subunits. And so these are really a family of cytokines that are very closely related, that have co-evolved together. And we think of IL-6 as being the sort of basal, the founding member of this family. And it's really important for a variety of inflammatory processes. So how do we ask whether these cytokines are important in the induction of CD8 T cell responses? Well, what we're going to do is I'm going to show you a variety of approaches that will sort of tease this apart. And just for the sake of time, I'm going to say that while IL-6 was the top rated cytokine or elevated the most, it's actually not required at all for the CD8 T cell response. And neither is IL-12. This was our top candidate for the cytokine that would be so important in driving the CD8 T cell response, and it's not important. So that's going to leave us to focus on IL-27. So how do we study this T cell response, the ability to generate a CD8 T cell response? And we use the COVID mRNA vaccine, and we use the influenza mRNA vaccine. But as a basic immunologist, the tool that we love most is the use of ovalbumin, which is a model antigen, which has been used by the immunology community for more than 50 years now to study the immune response. And because of that, there's a wide variety of tools that we can utilize to help understand how things work. So for example, there are OT1 T cells, which are TCR transgenic T cells that respond to OVA. We can make an LNP that contains OVA mRNA. So instead of using a COVID spike protein, we're using the OVA mRNA, so ovalbumin mRNA as an antigen. There are also T cells that are CD4 positive that recognize this. And then we have tetramers to be able to identify the cells that are responding to OVA. We have an antibody that recognizes the OVA class 1 complex, so we can see which cells are presenting antigen. And then, of course, we have a peptide that we know that these T cells recognize, and a peptide pool that we can use. So we have a really lovely array of tools to be able to say, once we vaccinate, how does the CD8 T cell response, how is it induced? How does it change over time? So this is a very simple experiment. So all we've done, we've ruled out IL-6 and IL-20 and IL-12. I don't have time to show you those data sets. So we're kind of focusing on IL-27. And this cytokine is a heterodimer composed of EBI3 and P28, shown here. And this is its receptor subunits, GP130 and IL-27. So all we're 27 receptor alpha. So all we're doing here is taking mice, B6 mice, or IL-27 deficient mice. We're giving them 1,000 OT1s, and the OT1s are these T cells, these CD8 T cells that recognize Synfecal. We're vaccinating them, and then over the course of 12 days, we're just bleeding the mice, and we're just going to try and quantify the T cell response to this vaccination. So this is I'm going to just show you the slide, and I'm going to let you interpret it. I'm going to let it rest for a bit. It's really a very simple slide, and the interpretation, I think, is pretty straightforward. So all we're looking at here on the x-axis is days post-immunization. So we vaccinate at day 0, and then we bleed the mice day 6, 10, and 12. And what we're looking at here on the y-axis is the percentage of the CD8 T cells that are OT1s. So in wild-type mice, when we vaccinate, we get a great T cell response. I mean, this is a terrific T cell response to a single vaccination. It peaks, and then as the immune responses want to do, it declines with time as antigen goes away. So this is a classic immune response. In the absence of either IL-27 or EBI3, there's really a paucity of a response. So we've taken away this one cytokine, IL-27, and the CD8 T cell response is awful. The humoral response is largely fine as far as we can tell, but the CD8 T cell response is really awful. So this just says IL-27 has a role in the induction of this T cell response. What it doesn't tell us is where IL-27 is acting, right? It just says that if we take the cytokine away, the T cell response is awful. The 27 receptor is expressed in a variety of different cell types. And we sort of became interested in like, well, okay, was this because IL-27 acts directly on the T cell, or is it doing something else? So to address that question, what we did was we took OT1 T cells and we bred them, those mice, those OT1 donor mice, we bred them to a mouse that lacked the IL-27 receptor. So now we've got OT1s that are either wild type or 27 receptor deficient. One T cell can respond to 27 and the other cannot. And then what we do is we mix these at a 1-to-1 ratio. We have a way of distinguishing these different populations. We mix them at a 1-to-1 ratio. We put them into B6 mice. We vaccinate, and then we bleed them again, and we look at the relative levels of these T cell responses. Do these T cells, whether they have the 27 receptor or not, do they expand at the same rate as those that are 27 sensitive? And again, we get a really, really sort of distinct result. So all we're doing here is taking away the 27 receptor, right, from these OT1s, where you mix them at 1-to-1, and at day 0, they're there at a 50-50 ratio. You vaccinate, looking at different days post-infection, and this is a great OT1 T cell response, but in the absence of the 27 receptor, these cells are really at a huge competitive disadvantage. So all this tells us is really that 27 is directly impacting the CD8 T cells, and we know they express high levels of this receptor when they're activated. So this just tells us that 27 is going to be really sort of key in acting directly on the CD8 T cell, right? Very, very simple interpretation. It's the most parsimonious explanation. So we've taken away IL-27. And usually if this was a live audience, I would then ask, well, what should we do next? But it's not a live audience. Well, you are live, but we're not particularly interactive today in this sort of setting. But the next thing to do is, like, we've taken it away. Well, what happens if you give it back? And there's a variety of options to give it back. We could simply give back a recombinant protein, or we could use a viral vector to overexpress 27 and see if that restores it. But the strategy we chose to use was instead of using over mRNA, instead of having an immunogenic protein, what we did is we took IL-27 mRNA and encapsulated that in a lipid nanoparticle. So essentially, we're using this platform to deliver a cytokine and have it produced in the lymph nodes at the site of action where priming is occurring. And essentially, what I'm showing here is the sort of the construct. It's the EBI3 and IL-27 subunits with a linker. So it's a single transcript, a single mRNA transcript. We include this mRNA in the lipid nanoparticles. And what I'm showing you here is bone marrow-derived macrophages given these lipid nanoparticles. So these macrophages are from an IL-27 deficient mouse. They can't make IL-27. And what we're doing here is we're staining for IL-27. And sure enough, these IL-27 knockouts don't make, in the absence of lipid nanoparticles, they don't make IL-27. If we give them an irrelevant lipid nanoparticle, they don't make IL-27. If we now titrate in different doses of the lipid nanoparticle, we can get all of these cells to make IL-27. We can measure the protein and secreted protein. This is a really, really efficient way of delivering mRNA to macrophages in particular, and to remodel them to make a cytokine that they normally wouldn't remodel, they normally would not be making. So what happens if we take these IL-27 particles and then mix them with over LNP? Will that restore the CD8 T cell response, or is something else going on? So just let me walk you through this. Over here, we have a B6 host that's vaccinated with LNP over. And in this case, we're not using OT1s. We're looking at the endogenous response that's generated. We don't need to use a TCR transgenic. We're using a tetramer as well to identify these cells. Actually, I think these are OT1s, but we see the same thing with endogenous responses. So here's the B6 host. This is I think about day 10 post vaccination. Here's this great CD8 T cell response. In the absence of the IL-27, we vaccinate, and then we have this terrible response. And here we're using either P28 or EBI3 knockouts. It doesn't really matter which one. We have this terrible response. And then when we give back IL-27, we clearly restore the response. So this just says, we can give back a cytokine. We can have it produced in the lymph node at the place where the CD8 T cells get primed and activated. And it really does restore the CD8 T cell response. So we're quite pleased with that. So it just says endogenous 27 is important, but we can also use this platform to sort of test that. Now, at the same time as we're doing these studies, you don't do these studies in a vacuum. At the same time, other people are interested in this very question. And there's a paper from Bali Palundran's lab, I think in 2022. And they sort of were one of the first labs to really look in depth at how the CD8 T cell response is induced in response to infection, in response to the mRNA vaccines. And there's a ton of, it's a fantastic paper. And one of the things they identified in their paper was that type 1 interferons had a role in inducing the CD8 T cell response. It's a really neat observation. And that's kind of like a funny observation in the context of what I've just told you. I've told you IL-27 is important. And these investigators said that type 1 interferons are important. And that's kind of like, oh, well, I wonder which one is that related? Are they important to understand their interactions? And because we've worked on IL-27 for a very long time, as Eleanor talked about, I happen to know that type 1 interferons are really good at inducing IL-27. That's in our literature. So for example, these are taking bone marrow-derived macrophages, and we give them either PBS or empty LNPs or mRNA or lipid nanoparticles that encode a pseudouridine-modified mRNA, and it induces IL-27 in culture. But if we block type 1 interferons by blocking the receptor, that's abrogated. And through data sets, I'm not going to bother, I don't have time to show you today. We know that in the type 1 interferon receptor knockouts, in fact, when you vaccinate them, there's a defect in their ability to make IL-27, which is what we see here. So we started to think that maybe part of our model was that, the vaccines induce type 1 interferons, and that's what drives IL-27. And so one way of sort of addressing that, one piece of data that I think helps us to think about that, is illustrated here. So here we're looking at the endogenous response using a tetramer to the ova synthecal peptide. So here's a wild type mouse that's vaccinated. These are the ova specific CD8 T cells. They're CD44 high, tetramer positive. And here are the type 1 interferon receptor knockouts. So essentially replicating what the Palundrum lab had reported. This is really very deficient. But if we give back IL-27, well, actually, you can see that, under some circumstances, this IL-27 can potentiate that CD8 response, and it completely abrogates any need for type 1 interferon receptor signaling. So we're sort of bypassing, we've done a runaround the requirements for type 1 interferon. So we're sort of developing a model, I think, of how these events work, and that's kind of illustrated here. And again, I don't have time to go into all the details. I'm happy to answer questions about this. But we're starting to think that these LNPs, instead of this being a pathogen, these lipid nanoparticles that contain mRNA are taken up by a cell. We think in the lymph nodes, it's mostly macrophages. These cells respond to the LNPs and make type 1 interferons. And then data I haven't shown you today, it's not these cells that make type 1 interferons, rather it's inflammatory monocytes that come into the tissue after vaccination, and they respond to type 1 interferons, and they make IL-27. And that IL-27 is required for the CD8 T cell expansion. So we sort of developed a model of like there's a division of labor. It's not just the cell that takes up the lipid nanoparticle that mediates everything. In fact, the sort of adjuvant properties of the lipids, help to bring in other inflammatory cells, as it would for an infection, for example. And those are the cells that really are important for driving this cytokine and providing signal 3 that's required for the CD8 T cell expansion. And the other thing that's kind of interesting here is that these LNPs are not very good at inducing IL-12, which is why we think that they're not so critical. But this is a cytokine that's not induced to high levels. It's not really critical for this induction of this T cell response because the LNPs don't actively induce IL-12, sorry. So in that context, we started to turn the question around a little bit and sort of ask, well, we can deliver IL-27 mRNA and it works really well. Well, what else can we do? Can we use this platform in a different fashion to sort of mess with the immune system to see if we can sort of violate the normal rules of the immune system? And I've told you that, that the LNPs don't typically induce IL-12. So we thought, well, if IL-12 is such a good inducer of CD8 T cell responses, what would happen if we gave IL-12, not protein, but if we encapsulated IL-12 mRNA in particles and then mixed it with the ova mRNA particles and then vaccinated? Would IL-12 matter, or would the effects of IL-27 trump those activities of IL-12? And so really the experiment is like, can we use IL-12? It's not induced normally, it's not part of this endogenous response, but can we override that biology and give IL-12? And again, the mRNA particles are really good at inducing if they encode the IL-12 mRNA, they're very efficient. They induce high levels of transient IL-12 for like a space of maybe 48 hours in the lymph nodes. And then when we vaccinate, we get quite a remarkable result. So let me just sort of walk you through this. So essentially here, the open circles, we're looking at time against the CD8 response to ova. Here's the open circles, and this is just what happens when we vaccinate with ova. And this is really, so a single vaccination. This is a terrific CD8 T cell response. It really is. It's a single vaccination. You get this peripheral CD8 response. You get long-term memory. We know it's protective against other rechallenges, and I haven't shown you that data set. But when we include IL-12, this is a good response, and this is an awesome response. We were like, oh goodness, this is really a terrific response. And that magnitude of that response doesn't just kind of go away in terms of as a response contracts. It leaves us with a larger memory pool in tissues, for example, that we don't normally generate with just a single vaccination. And so there's lots of ways of thinking about this and lots of things that we can do with this approach or utilize this approach. But one thing we did, one model we used was sort of in the context of a cancer vaccine, in the context of a therapeutic vaccine. And the next slide just illustrates that. And it's going to take a little moment to work our way through this slide. Essentially, because immunologists like ova, we have access to a variety of tumor cell lines that express ova. And here we're using a B16 melanoma that expresses ovalbumin. And it's just really a model to test if we can augment the immune response to this tumor to the ova, what will happen. And essentially, this is a very aggressive tumor. And if you put it into mice, the tumor grows really rapidly, and then it reaches a large volume, and the mice have to be euthanized because the tumor's really large. Now, what we've done here is we've used a model where it's a sort of therapeutic vaccination. So you give the tumor, you wait a few days, and then you give a primary challenge with ova or ova mRNA or IL-12 mRNA, and then we give a secondary challenge round about day 11. Okay, so you get the cancer in, you let it establish, and then you give these two challenges. Okay, and PBS, you know, no saving them. You know what? IL-12 alone, if we just use IL-12 mRNA, IL-12 is really a very potent cytokine. Alone can provide quite significant protection. If we use ova, modified pseudouridine modified ova, there's a delay. There's a delay, but it's not terrific. But when we use this combination of the modified ova and IL-12 mRNA in this sort of therapeutic setting, you know, we completely block tumor growth. So this is sort of an example of a therapeutic vaccine. And some of you may be aware of the paper that just came out in Nature yesterday that's a follow-up to the pancreatic cancer mRNA vaccine showing efficacy there. So we think this type of approach will be useful or applicable to thinking about therapeutic vaccines in that context. And then I think sort of my final slide is really a sort of a call to arms to think about how we can use lipid nanoparticles and delivery of mRNA to modify these cells. We've got them to make IL-27 and IL-12, and we have a whole bunch of other cytokines that we've engineered them to produce. But the other thing that we could start to think a little bit about is just like if we were engineers, you know, what would we want these cells to do? Not just for CD8 T cell expansion, but for other factors. And as immunologists, most of us will be aware that there's signals 1, 2, and 3, TCR, co-stimulation, and signals 3 mediated by cytokines. I think we have the opportunity to think about what payloads, what mRNAs do we want delivered to these cells that might change their phenotype, their function. You could think a little bit about tolerogenic particles. You could think about different classes of T helper cells that you might want to induce. I think we have this really nice opportunity to think a little bit about that as a strategy going forward and utilizing this platform. My very last slide is really just sort of reiterating that. The modular design of the LNP platform, it's an immunoengineer's dream. I mean, I didn't show this, but we already used this response to enhance the cellular responses to COVID and influenza mRNA vaccines and tune the type of response and enhance and augment memory cell responses to pathogens, for example. And then the other thing, the last point I want to make is, you know, cytokines are often considered toxic. I've seen slides today where people talk about such and such a cytokine is pro-inflammatory. But cytokines are really just proteins trying to do a job. They're not necessarily pro or anti-inflammatory per se, but they are and can be associated with toxicity. And I think we can think about ways of using this technology to bypass that toxicity. And I like to end with this quote. It's from Bill Paul, who was an immunologist at the National Institute of Health. And in Cell in 1989, in a commentary article, he was talking about lymphokines and whether we should think about these as therapies or targets for therapies. And one of the sentences he wrote, which has resonated with me for a very long time, is, surely, if lymphokines prove to be valuable therapeutic agents, and I think we all know that they are, new approaches to a physiological type of drug delivery, mimicking normal modes of action, will be needed to improve their efficacy and avoid their side effects, which are almost certainly to occur from systemic administration. And I think this approach, using these mRNA LNPs, sort of bypasses a lot of that, and I think maybe addresses this point that was made by Bill Paul. With that, I'm really done, and really just so important to acknowledge the individuals. I get to sit here today and talk about the work, but the work done by Anthony and Mohammed and Emily and a whole host of other investigators and collaborators that have made this possible is really, it's just, you know, when we work together as a group of scientists, you know, we can achieve an awful lot. And maybe that's just a take-home message to remind everybody today that we can still do that. With that, I'm happy to take any questions if there are.